Hello, welcome to our third lesson in our third subtopic on genetics. We are going to look at genetic crosses. So this is our lesson 5.3. But before we get into what we have for this lesson, let us refresh our minds on what we did in our first lesson and our second lesson. In our first lesson, I talked about some important terms that are used in genetics. So I talked about the gene, I talked about locus, alleles, whether they are dominant or recessive, and I also talked about genotype and phenotype. Please, if you haven't watched our first video, do well to watch it before you join us for this particular lesson, because a lot of the terms that were described in that particular lesson are going to be used in our lesson today as well. Then in our second lesson, that's lesson 5.2, I talked about Mendel's experiments as well as the laws of inheritance. So I talked about Mendel using 7 traits of the pea plant to experiment or to find out how traits are passed on from parents to offspring, and the conclusions that he also came up with. So with the laws, I talked about the law of dominance, I talked about the law of segregation, and I also talked about the law of independent assortment. But in that case, I gave the exception to that particular law. So some people even refer to these laws as principles, yes. So I mentioned them in our second lesson, that was lesson 5.2. So please, if you haven't watched lesson 5.1 and lesson 5.2, do so before you join us for our lesson today. We are going to look at genetic crosses. But before we get into it, please, for you to be updated on all our videos, remember to subscribe to our channel, because we'll be releasing our lesson videos from time to time so that you won't miss any of them. Our lesson 5.3 on genetic crosses will be in parts. So this is going to be the first part of it, and we're going to concentrate on monohybrid cross. So this is going to be lesson 5.3.1. So the first of the lessons on genetic crosses. Then there will be another lesson 5.3.2, and there will be 5.3.3 and so on. They'll all be handling the various aspects of genetic crosses as I have segmented them. Right. But before we proceed, let us look at the objectives. At the end of this lesson, the student would be able to explain genetic crosses, and the students should also be able to illustrate monohybrid inheritance in genetic diagrams. And as well, the student would be able to explain true breeding. So now let us move on to look at what genetic cross is about. Genetic crosses are used to determine the possible phenotypes that would show in offspring. We are talking about inheritance, and we know that inheritance is the passing on of traits from parents to offspring. So, one way that we would be able to predict what kind of phenotypes would be observed in the offspring is by the use of genetic crosses. So there are parents, or in the 2 individual parents that are going to be crossed, and they possess certain traits. Now, how are we able to tell whether this particular trait would show in the offspring, or that particular trait would rather show in the offspring? Yes, we cannot just use intuition to tell. We would have to do genetic crosses. So that will guide us into knowing exactly what phenotypes would be shown in the offspring. So that is what genetic crosses would be for. Now, there are types of genetic crosses. We can talk about monohybrid cross, which is what we are going to do today. And then we talk about dihybrid cross. Well, when you compare these two, mono talks about 1, di talks about 2. So monohybrid cross would mean that we are going to do some crosses that involve 2 individuals that differ in 1 character. Or the trait that we are considering in this case will be just 1 trait. So that is what we refer to as monohybrid cross. But dihybrid cross would mean that you are going to consider 2 traits. And there will be back cross and test cross, and we're going to use them to tell whether a certain phenotype which is being observed in offspring is or are homozygous or heterozygous, or the organisms involved are homozygous or heterozygous for that particular trait we are talking about. Yes. Don't worry about the terms that I'm using, because I'll be explaining them very soon, and they are nothing to worry about. Okay. So let's move on to some important things that we need to know. I've already talked about this. That's the first thing for us to note, that Monohybrid cross is a genetic cross between two individuals that differ in one trait, or a cross in which only one trait is being considered, and I know that I've talked about that. In monohybrid crosses, usually two purebreds or true breeding organisms are considered. And what do I mean by this? When you talk about purebreds, we are talking about organisms that only produce the same kind of trait. For example, if you consider a group of plants that produce purple flowers, so the plants produce purple flowers, the parents that produce those offspring with purple flowers were also produced by parents with purple flowers. And those parents were also produced by parents with purple flowers. It means that when you consider the lineage, they only produce purple flowers. So that makes them purebreds. Those organisms that were produced from the group are all purebreds. So we also say that they are true breeding organisms for that particular trait we are considering. You can also have another group that only produce white flowers, and apart from white flowers, they won't produce any other color. So that makes them also purebreds for that particular trait, or true breeding organisms for the trait. Right. So, as I said, we are going to use purebreds in our monohybrid crosses. When we do these crosses, the first offspring that are going to be produced from our cross would be referred to as the first filial generation, or F1 generation. So this is very simple to keep. When we do the monohybrid cross, or any other cross that we're going to do, the first offspring that are produced from our cross would be referred to as the F1 generation. Now, when we use this F1 generation to also produce another offspring, then that is going to be the F2 generation. So it's just like parents producing children and the children also producing children. So those ones become the grandchildren. So let us note that point. Parents produce F1 generation. The F1 generation would produce the F2 generation. Let us also note that true breeding organisms are homozygous for their traits, and I'll be explaining the term homozygous very soon. But note that true breeding organisms are homozygous for their traits, but when you cross them, they will produce offsprings that are heterozygous. So it means it is very important for us to understand these terms, homozygous and heterozygous, and they are very simple. Homozygous organisms, or homozygotes, and here you can tell that homozygous, G-O-U-S, means that is the adjective form, and homozygotes, G-O-T-E, means that is a noun. So homozygous organism, or a homozygote, has two identical alleles for a particular gene. So for the trait that we are considering, yes, if the set of alleles that make up the genotype for the trait we are considering are the same, then it makes that organism homozygous. So for example, if the trait we are considering is, say, color of the flower, or the height, or the type of blood, and the organism has the same kind of alleles. So for example, an organism has capital A, capital A, capital B, capital B. You see that the letters are the same for the genotype. Capital X, small letter A, small b, etc. These tell that the alleles are the same for the particular genotype we are referring to, and so it makes that organism homozygous. But heterozygous, on the other hand, would mean that the organism would have different alleles for a particular gene we are considering. So, for example, if the genotype is capital A, small a, capital B, small b, capital X, capital Y, capital A, capital B, etc. These are all examples of heterozygous organisms, or heterozygous as in the case that we are considering here, meaning that the alleles are not the same for the genotype. Okay, so now you know what F1 is, you know what F2 is, you know what homozygous means, you know what heterozygous means. So we can now start by looking at our monohybrid cross, which is what we want to do in this particular lesson mainly. So let us take an example. Example 1. We are going to do a cross between two pea plants that differ in color of their flowers. And the colors we are going to consider here are purple and white. So I'm saying that where allele for purple color is capital A and is dominant, and allele for white color is small a and is recessive. And we are to note that they are true breeding for the traits that we are coming to talk about. So true breeding means that the purple-colored parent would be homozygous for the trait, and a white-colored parent would also be homozygous for the trait, meaning that their genotype would have the same kind of alleles. Alright, so now let us move on to look at them. We start with the parents. Here they are saying that one of the parents produces purple colors only, so like this, purple-colored flowers, and the other parent produces white-colored flowers. And since they are true breeding, it means they are homozygous, meaning their alleles are the same. So here it is saying that purple color has allele capital A. So if purple color has capital A and it is true breeding, meaning that it is homozygous, then it means the allele should be capital A, capital A. And it's crossing with another which has white flowers and is also true breeding. But white flower gene is small letter A, and since it is true breeding, it should also be homozygous, and that means it will have small letter A, small letter A. Now, for us to have a better cross, it is very important for us to separate the genotype into alleles, because when these parents are crossed, their gametes are going to be mixed. And remember that since their gametes are going to be mixed, it is also telling us that both alleles wouldn't be sorted into the gametes. And I explained this in our second lesson. Yes, there will be segregation. And so only one of these should be taken at a time into the gametes. So either that offspring will choose this one or will choose that one. The offspring cannot choose both. So you should choose one from this first parent. And you should choose one also from this second parent. So let us look at the possible combinations for the F1 genotype. It is either the offspring would choose the first one and the first one. So the first one from the first parent and the first one from the second parent. So first to first. And that is going to give us capital A, small a. Or first to second, like this, and that is going to give us capital A, small a. Or there is a possibility an offspring would choose second to first, like that, and that is also going to give us capital A, small a. Or second to second, and that is going to give us capital A, small a as well. But now let us look at something. You can see clearly here that the offspring are having the same kind of genotype. Yes, because the parents were having the same kind of alleles themselves, and so they would only give one of them. So whichever of them that would be given, for example, the parent with capital A can only give capital A, and the parent with small a can only give small a. So the offspring would end up having only capital A and small a as their genotype. And since capital A is for purple color and is dominant, it will make all of them purple colored. So like we see in this particular phenotype, all purple colored flowers. But note that they are going to be heterozygotes. Yes, heterozygotes because although they are purple colored, but they have one allele for purple color, one allele for white color. But purple color is dominant, and so it will show itself on the outside. So that is why phenotypically they are all going to be purple colored. You understand these terms that I'm using very well if you have watched our earlier lesson videos. So I said that if you haven't watched our lesson video 1, that is the 5.1, and our lesson video 2 on genetics, that is 5.2, please watch them before you proceed with us on this particular lesson. Yeah. So this is how it's going to be. Now let us look at what will happen in the case of the F2. So we are coming to pick the F1 and we'll cross them. Here we know that the F1 genotypes are all capital A, small a's. So we are going to pick any two of them and cross them to see what will happen in the case of the F2. So with the F1 phenotype, there are purple colored flowers crossing with purple colored flowers, but remember they are all heterozygous for this trait. So their genotypes would be capital A, small a crossing with capital A, small a. Now let us separate them into their gametes because both wouldn't be chosen. Only one of them will be chosen at a time. So capital A, small a crossing with capital A, small a. Right. So it means that the offspring that are going to be formed would have to choose, in this case, they would have to choose only one of this. They can't choose both, so they will choose only one of this from this parent and would also choose one of this from the second parent. So let us look at the possible combinations. F2 genotype. I said that the offspring that are formed from the F1 are called the F2. So F2 genotype. So the possible matches could be the first with the first. So the first from the first parent and the first from the second parent. And that is going to give capital A, capital A. And let me remind you that here you can see that this allele is being shown with a different letter, indicating that although this parent was purple colored, it still had the white colored gene inside it, or the white colored allele inside it. That is why we are seeing it here, just like the case of this one. Okay, so now we have looked at the first possible combination of alleles to form the offspring. So capital A, that is when the offspring would choose the first from the first parent and first from the second parent. But what if the offspring would choose the first from the first parent but second from the second parent? And that is going to give capital A, small a. Well, the offspring may also choose second to first. So second from the first parent and first from the second parent. That is going to give capital A, small a. Otherwise, the offspring would choose second to second, and that is going to give small a, small a. So now we are observing something different in here. Yes, let us look at it here. You can see that this one is capital A, capital A. Capital A is for purple color, so it means this one would be purple colored. Capital A is present here also, together with small a, but capital A is dominant, and so this would also be purple colored. And then the same thing happens here. But interestingly, this one is going to have only small a's. Right. Let's understand it from this point. It doesn't mean that the first offspring that will be produced would definitely have capital A, capital A. No. This is only giving us a ratio or probability that one of them probably would choose capital A, capital A. Then you have two of them choosing capital A, small a. And you have one of them choosing small a, small a. It is just probability. It could even happen that the very first offspring to be produced may choose this combination, and maybe the second offspring would choose this combination, the third would choose this, in that way. And even depending on how many of the offspring were produced, the order may even change. So this one is only telling us how the combinations would be, not necessarily the order in which the offspring are going to be produced. No. Okay, so let us look at the phenotypes. So for the F2 phenotype, we expect that there will be 3 purple colored flowers, and 3 individuals that produce purple colored flowers, and there will be 1 individual that would produce white colored flowers. But like I said, we are talking about ratio in this case. So it means that we are not saying the parents are, or the F1 that were used as the parents in this case, are going to definitely produce 4 offspring, where 3 of the individuals would have purple colored flowers and 1 individual would have white colored flowers. No, that is not the point. It's ratio. So we are saying that assuming the parents produce about 12 offspring, out of these 12 offspring, we expect that 9 of them would have purple colored flowers and 3 individuals would have white colored flowers. If the parents produced 24 offspring, we expect that 18 of them would have purple colored flowers and 6 of them would have white colored flowers. Right. If they produced 40 offspring, we expect that 30 of them would have purple colored flowers and 10 of them would have white colored flowers. So it's just ratio. So it would be numbers around that, or approximately that. So let me say so. Okay, so this is our very first example, and I know that you have enjoyed the first example. We have looked at true breeding parents that produced the F1, and we have used the F1 to produce the F2. And we saw the case where the true breeding parents produced offspring that were heterozygous, but they were all purple colored in terms of their flowers. But when the F1 were crossed, they produced F2 that showed some having purple colored flowers and some having white colored flowers in a certain ratio of 3:1. Right. So it means that when the true breeding parents are crossed, only the phenotype of the dominant allele would show. But in the F2, the recessive one will reappear. So although in the F1 we didn't see any white colored flower offspring among them, but in the F2 we are seeing it. So that means it was hidden in the F1, but it has reappeared in the F2. Alright, let us now move to look at another example so that they will help us improve on our understanding on this concept. Example 2. In a certain plant species, round shape of seed is determined by a dominant allele capital R, and wrinkled shape is determined by an allele small r. What phenotypes would be observed in the F1 and F2 if a true breeding parent with round seeds is crossed with a true breeding parent with wrinkled seeds? So here they have made us aware that the round seed is controlled by an allele capital R and is dominant, and the wrinkled seed, of course, would be recessive and it is small r. Yes, so now they want us to do a cross between true breeding parents for both. So true breeding parents for round seed and true breeding parents for wrinkled seed, and find out what would be observed in the F1 and the F2. So now let us look at the parental phenotype. Round seeds against wrinkled seeds. So they are true breeding. It means that they are going to be homozygous for the trait. So parental genotype capital R, because it's homozygous, so the same kind of alleles. Crossing with small r, that is also homozygous. So let us separate them into their alleles, or how they'll be found in the gametes. Capital R, crossing with small r. Okay, so now let us look at the possible combinations in the F1. So the first would give capital R, small r. First with second would give capital R, small r. Second with first would give capital R, small r. Second with second would also give capital R, small r. So in this case, we are going to observe that the F1 phenotype would show only round seeds. But let us also remember that although they are showing round seeds, but they are going to be heterozygous. But they will show round seeds. So it means in the F1, the wrinkled seeds would be missing. Now let's look at it if they are going to reappear in the F2. So then let's see the possible phenotypes in the F2 generation. So the F1 phenotype, we only got round seeds. So two of them would be crossed. Let's look at their genotypes. Capital R, small r, crossing with capital R, small r, because that is what we saw in that case. And now let us separate them into their gametes or their alleles, as how they would be in the gametes. So either this or that from the first parent, and either this or that from the second parent. How would the offspring choose their combinations? Yes, F2 genotype. It is either the first from the first parent with the first from the second parent, and that is going to give capital R, or first from the first parent, second from the second parent, and that is going to give capital R, small r. There is also the possibility of second from first parent, first from second parent, and that is also going to give capital R, small r, or second to second, and that is going to give small r. So just like the case of the color of flowers, you see we are finding a similar thing in this case. Now we are going to observe that in their phenotype, there will be 3 individuals with round seeds and 1 individual with wrinkled seeds. So what does it mean? Wrinkled seeds disappeared in the F1, but now it has reappeared in the F2 because the F1 are heterozygous, and so they had the alleles for wrinkled seeds hidden in them. So when you cross the F1, those alleles that were hidden would now be combined again. So the offspring that chooses small r, which was hidden in both parents, and combines them, would eventually have the wrinkled seeds. Right. So we are seeing that there is a possibility one would be homozygous for round seeds, and there are 2 that would be heterozygous for round seeds, and 1 that would have the wrinkled seeds. And I said that this doesn't show any particular order. It is only giving us probability. Right. So this is for monohybrid crosses, and it is very simple. and I know that you have enjoyed how monohybrid courses have gone today. Right. Okay. It will be our final example for our lesson today. So let us take a summary of what you have looked at. So far, I've talked about genetic crosses and their types. I mentioned monohybrid cross, dihybrid cross, back cross, test cross, and some of them I'll be explaining later. But I spent time talking about the monohybrid cross, especially. And I've also explained what true breeding organisms means, or purebreds, what it means. And I've talked about F1 and F2 generations. I've also talked about homozygotes and heterozygotes. Yes, where I said the homozygotes have the same kind of alleles for the traits we are considering, whereas heterozygotes have combination of the different alleles, but for the same gene we are talking about. Okay, so this is what we have for today's lesson. In our next lesson, we'll continue with genetic crosses. Just like I said, they are in parts, so we will talk about genetic crosses, but we are going to talk about a situation where we have the first parents that we are considering, at least one of them to be heterozygous. Right, so that is the situation we are going to have in our second lesson, and it is also going to be very interesting. But just like I do, I have these few questions for you to try, and I'll add an email address to this particular video, so make sure you check it in the comment section or the description section. You're going to see it there. So the first question: explain briefly what is meant by homozygote, heterozygote, true breeding. Question 2. A true breeding tall plant was crossed with a true breeding short plant of the same species. If allele for tall plant is dominant and is represented by a capital T, whereas allele for short plant is small t and is recessive, find the phenotypes that would be shown in the A. F1 generation, B. F2 generation. Alright. Yes, it's good. We have enjoyed the lesson today, which, yes, that is what I hope. So, we have come to the end of our lesson today. But, just like I say, please, for you to be updated on all our videos, don't forget to subscribe to our YouTube channel. Okay, so see you in our next class.